so I guess we'll get started with today's material. Before we get going, are there any questions anyone has left over from the last class? Any issues yet about genomes or where we get them or how we annotate them? Okay, so today's lecture will be getting much more into the practical hands-on issues of working with genomes. And there are limits to what you can do with just web available resources. So I'll try to give you pointers to where you can get information you could use for more sophisticated kinds of analyses. But we can get pretty far with the resources that are available to us and relatively easy to demonstrate here in the lecture. What I want to do is get into a bit of that material. There's not really a lot of background I want to talk about today before we get to that. So we have some material here I just wanted to mention about the status of whole genome sequencing, because this is one of the things that has really changed dramatically and is very quickly changing what is available to us and what we can do with computational resources. So I think when I started as a professor here, which is not that long ago, there was one vertebrate genome available, so there was human genome. There are now, at this point, almost 150 vertebrate genomes, over 200 relatively large eukaryote genomes, and thousands of prokaryotic genomes, including almost 30,000 partially assembled genomes, as well as almost 3,500 fully assembled, and over 4,000 viral genomes. And this is a phenomenal resource for doing all kinds of work that wasn't possible just a few years ago, from working with some of the ideas of comparative genomics and other applications of genome resources we've been talking about earlier in the week. So this is just to motivate why we want access to this kind of information. And what I mostly want to do today is go through some of the resources we can get to and ways of accessing them. I will say that there's not a lot that I would consider computationally deep about this lecture. It's more about where to find things and what kind of things you can find and how you can visualize them. But that's important to know. And I would say when you get into recitation or working on the assignments on your own, play around with some of these tools I'm showing you. See all the options and other resources I don't have time to go into, because there's much more there than I have time to show you. But are there any questions about any of this while I get to some of the actual resources? Okay, so I'm going to jump out of the slides here, and I'm going to start by bringing us to something I mentioned briefly before, that is the NCBI Map View, or Map Viewer, which is NCBI's standard genome browser. So this is a web page devoted to Map View, and what this has associated with it is all the different genomes that are integrated with Map View. Generally, this is not going to be the small genomes, so things like a bacterium, but we do have quite a few large genomes to choose from. So if you can see this, we've got 147 vertebrates, and as well as many others. And by clicking on these links, we can say that maybe we're interested in particular invertebrate genomes, and maybe particular mammalian genomes, and maybe primate genomes specifically. And if we're working on things that have more of a medical relevance, there's a pretty good chance that we're specifically interested in the human genome. So we can see that there are actually a couple of different human genomes available here. So this is referred to as different annotation releases of the genome. Really, this is probably the same assembly, but the annotation of it has been updated. So we've now got the 106th release of the annotation of the human genome. And this is something that will be updated periodically and will continue being updated. But we can click on this and we can see what goes into working with a genome. So this is a high-level overview of the human genome. You can see we've got our 23 we've got chromosomes 1 to 22 here. We've got the sex chromosomes, and we've got a small bit of mitochondrial genome here. And usually this isn't what you're going to be looking at. Usually you're going to need to zoom into a particular piece of the genome to find anything interesting. And I'm going to show one way of doing that by doing a search term. I'm going to type in GABPA, so that is a search we can use to find annotation on the genome associated, in this case, with that text term GABPA. So GABPA is a kind of gene, and what we can see here is that annotation to this pops up in the human genome in a couple of places. So it comes up here on chromosome 7. It comes up several places on chromosome 21. Now, if we look carefully at what is popping up there, we can see that there are a lot of kinds of annotation on this genome that may be matching that search term. So over here, we can see that there are a number of things that are annotated as type transcript. That means that there are RNAs that have been aligned to this genome that are showing up as a match to that search term. And on chromosome 7, we can see that there is a set of so that would be, in this case, mus musculus, so common laboratory mouse. Mouse RNAs have been aligned to the human genome, and the mouse RNA for this gene seems to align to a particular point on the human chromosome 7. We also have human transcripts available, and then we have other things that I'll point out in a moment, so other versions of this, other kinds of elements. But this is probably more complicated than we want here. So what I'm going to do is try filtering this just to genes. So get rid of all the transcripts and see just things that are genes aligning to the genome. And then the hits to chromosome 7 go away. Can anyone think of why we might lose the hits to chromosome 7? Why do transcripts align there but not genes? Well, probably what this is telling us is that there is a gene in the human genome called the GAPPA gene, and it happens to have a homolog on chromosome 7. So something else homologous to it is on chromosome 7, so the transcripts are aligning in both places, but the actual gene is only annotated on chromosome 21. Even on chromosome 21, though, we can see that the annotation is showing up at, looks like 3 different places on the chromosome. And to understand why that is, we can look at what is showing up in that annotation. So the gene is actually being annotated multiple times on chromosome 21, sometimes in different places on the chromosome. So these first 2 here are showing actually 2 different annotations of the gene. So gene seq would be NCBI's canonical representation of the gene. OnceGenes is another kind of canonical gene representation, so a curated data set called the ensemble genes. And those maybe have slightly different ways they've annotated it, or slightly different ways that maybe they're saying the canonical splice form of that gene works. So that's one of the reasons we see multiple hits to the genome. But we also see a couple of other things that are both listed as gene seqs, the NCBI canonical representation of this gene, and they're showing up in different places in chromosome 21. So can anyone figure out why that would be? Well, we can get a hint if we look at this column here, which is telling us that this is referring to different assemblies of the genome. So these are 2 older assemblies that were more special purpose kinds of things. So the human genome has been studied enough that there are now multiple assemblies. Different groups have tried to do research on sequencing and assembling the human genome, sometimes with different data sets, and they will end up with different assemblies. And in some cases, those assemblies can have quite large distinctions between them. It's possible that those represent actual variations between the genomes of different people. So this UREF was a genome of a particular person, and it may be that that person has a genome where things are somewhat differently ordered, but more likely it's a mistake in the assembly of one of the genomes. These are older ones. We'd say probably those have been less well corrected for these kinds of rearrangement errors. The one that's just called reference is what NCBI would put out as the standard reference human genome. It probably has mistakes too, but it's at least probably our best guess as to what the right assembly is. So you always need to be careful of that when you look at these. But let's just say we will pick the gene seq annotation on the reference one, and we will go there, and that will take us to a particular point on the genome where we can now see this particular gene. I'm going to scroll down here so we can see a bit of what's going on there. And what we can see is this GAPPA gene. This is a model of the introns and exons. So the thin bars are intron, the thicker regions are exon. And then we have the name of the gene and a lot of different links to various tools we would have available for working with that gene or working with genes in general. I'm going to demonstrate some of these. Others will come up later in the term, and others you can just play with them yourselves and try to figure out what they're good for. but we have that available, and we can see where that sits relative to various other genes in this region of the genome, and get some basic information about gene order, gene arrangement, and potentially other signals around the genes. Are there any questions yet about any of this? All right, so some of these tools are things that are pretty handy to have if we're working with these kinds of genetic information. So sometimes you want to zoom in with this SV thing to what's called the sequence view level, so to get a much finer view, a different kind of genome browser that is really tuned for looking at things on relatively small scales. It has much more limited kinds of annotation available to it, but this is useful for looking at the scale of a single gene. And if we really want some detail there, we can zoom in much more here, maybe scroll to try to find a particular exon within that gene. I guess they're located. And once we found something of interest, we could maybe zoom to the base level, and we could see in detail what the nucleotide sequence is, try to find information about, for example, the splice sites, if we were interested in splicing, whatever else we were interested in. You can also get additional information by clicking on these to see the transcript models, the proteins, the individual codons, and all of that. So when you really want to zoom in and get fine-scale detail about a particular region of the genome, this is a handy tool for doing that. Any questions yet about any of this? Okay, so that is one thing that is available to us. Another tool that's pretty easy to use but makes some of the analysis we did in more complicated kind of manual ways earlier in the term a lot easier to do is this DL or download link. So if we want to download the sequence of this gene and maybe we're interested in regulatory signals around the promoter, we can say that we'd like to add an extra 2K around the gene and then say that we are interested in displaying this or downloading it or whatever, get exactly the piece of sequence we want out of the genome. So if you're trying to retrieve gene sequences, and especially if you're interested in things like the signals in the promoter region, this is, again, a really handy tool to have. Take some of the stuff that we did earlier without this and makes them a lot easier to do. So one more thing I wanted to briefly show you before we move on to other issues here is, if I go back here, this EV, or evidence viewer. So I've tried to convey over and over here to be careful about what the evidence is behind your informatics inferences. When you see something like a gene annotated in these map viewers, you're seeing a model of the gene that may or may not be the correct spliced form of the gene. It might have mistakes, it might be combining different splice forms into a kind of consensus model. So if you really care about a gene of interest to you, you want to be able to look at the evidence behind the annotation. And that's what this is showing us. So what this is showing is that there are various transcripts that have been aligned to this gene, and these are giving us the evidence that supports the canonical gene model in blue at the top here. And if you look carefully, you can see that in this case, this does seem to be pretty much a well-supported gene model. We've got these thick bars are all the exons in the gene, and we can see that more or less, aside from the start and ends, the 5' and 3' regions, we basically have the same conserved exon structure across every transcript known for this gene. Now, that doesn't mean there isn't some alternative transcript that isn't in the databases, no one was looking in the right condition or the right time point. But here we could look at this and we could say this is pretty strong evidence that this is a good gene model, that all of these exons at least do occur and do occur at least a large fraction of the time in that gene. We do see very often differences around the start and end of the gene. Those may reflect genuine variation. So often the start of a transcript is not entirely conserved, or they may reflect mistakes in the sequencing process, so just pieces of the gene get cut off in different transcripts. We also have additional information from ESTs, those short pieces of sequence I mentioned, and here we can see there may be some weak information for possible alternative exons. So maybe ones that are found in rarer splice forms. So we can see, for example, there's a piece of sequence here that shows up in an appreciable fraction of these ESTs. So it's coming up as something transcribed a good part of the time. It's not in any of these transcript forms. It might mean that it's transcribed for some different reason. Could be that there's even a little RNA gene hidden within this bigger gene, or it might mean that there is a rare alternatively spliced cassette exon sitting at that point in the genome. But these are all questions we would want to pay attention to if we were really trying to study this gene in depth. So any questions? Okay, so that shows us basically the tools available to us. But I want to show in a bit more detail some of the options available for working with a genome like this. And what I'm going to do is use this icon at left here to zoom out to the scale of the entire chromosome and try considering the issue of annotation tracks. So I've mentioned a few times this notion of tracks of annotation of a genome, and that is really what you want to work with when you're using a genome. You want to be able to take different pieces of information. If we click on this Maps and Options tab, what we end up with is this tool that lets us see the kinds of annotation track that are available and maybe add some ones of interest to us. So we have various ones related to different kinds of sequence. I'm going to close that. We have cytogenetic maps. I'll just show a couple of examples of these. So I will put in what's called an ideogram map, and I'll put in this Middleman breakpoint map. You can verify that those have been added to the tracks to be displayed. Click OK here, and then what this should show us is a couple of additional tracks of annotation added to our genome view. So one of the things we are seeing here on the left is this ideogram. And the ideogram, we can see, is showing us a model of the chromosome, and then we have these streams of information here, like 21q, 21.1. And you can notice, if I scroll to the right here, that those are often annotated with these genes. So we will see that a particular gene says 21q, 21.3. So this is a way that people traditionally localize things on the genome that was, in some ways, I'd say it's almost obsolete now that we have actual genome sequences. But in the pre-genomic era, you needed ways to figure out which things were in which places on the genome, and you couldn't do it at as fine a scale as is done primarily today. And these ideograms, so this kind of location is what's called a cytogenetic map or a cytogenetic coordinate, and it's giving us information about where things are found in the genome. And this is still used quite a bit in some contexts. So I did want to stop for a minute and explain how to interpret these, because it can be confusing if you haven't seen this kind of code here before. All right, so every human chromosome, or well, not everyone, but our autosomal chromosomes will come in the form of pairs of chromosomal arms, and there will always be a short arm that's known as the P arm and a long arm that's known as the Q arm, and those are separated by the centromere of the chromosome. And we would say that if this is chromosome 21, anything on the P arm we would say has location 21P, anything on the Q arm has location 21Q. And these cytogenetic coordinates are a kind of hierarchical way of going to finer and finer levels of detail. And you will often see them at different levels of detail in different applications. So it might be that chromosome 21 just has one level of detail on the P arm. So we would say that that has the subdivision 21P1, whereas maybe 21Q has two subdivisions that we would refer to as 21Q1 and 21Q2. Within one of these subdivisions, we may have a finer subdivision. We have that 21P1 is maybe broken into 21P 1.1, 21P 1.2, and 21P 1.3. And those may have further subdivisions. So maybe 21P 1.1 is broken down into 21P 1.11, and 21P 1.12, and maybe 21P 1.2 is broken down into 21p 1.21, and 21p 1.22. So that's basically how you read these kinds of coordinates, and each digit you add is breaking it down to a finer level of detail. So I mentioned this in part because this numbering can be confusing if you don't know that, so it may seem kind of odd that 21p12 is followed by 21p21. That's the reason those are likely to be consecutive in the sequence. You'll see things like that. So just be aware when you see terms like that. So if we refer to a gene as being at, let's say, in the GAPTA case, 21q 21.3, that's what it's referring to. And if we were really interested in looking in detail at that, we could go to the chromosome model here, we could pick 21q 21.3, and we could zoom in on that to try to see more specifically the genes that are located in that particular region of the genome. So are there any questions about how to read these things, or what I mean by cytogenetic coordinate? All right, so one thing to be aware of here in these cytogenetic coordinates is that this is very species specific. You'll see these kinds of coordinates for human beings. It will be done a different way in any other organism. So your traditional experimental biologist usually doesn't work on a lot of different organisms, so it's fine if every organism has its own way of doing things. For those of us who are computational biologists, we generally work on a lot of different organisms because it's all sequenced to us, and then it becomes kind of an annoyance that every community does things its own way. But just be aware of that. Some of these things, there are good reasons for every community to kind of create its own way of thinking about things. All right, so any questions about any of that? All right, so I want to give a bit of an idea then about this issue of different communities by going to a different organism here. What I'm going to do is go back to Map Viewer homepage, and we'll close the mammals here, and I'll look at invertebrates, and I'll pick an insect, and I will specifically pick an important insect model organism, Drosophila melanogaster. So here we have the fruit fly. That's one of the really well-studied model organisms for a lot of contexts. And there will be a fruit fly genome annotated in these genome tools. Fruit flies have a much smaller, more compact genome than human beings. And we can look for things on this genome. I'm going to pick an advanced search here because this will sometimes make things a bit easier to search for. I'll type in the name of a gene. So RPS5A. And to make things a little easier to search for, I'm going to clear all of the types of objects you could look for except for genes. And then I click Find, and hopefully that will turn up just the genes annotated RPS5A, and there's just one gene called that, so that will bring up a result that is on the Drosophila X chromosome. So we can pull that up, pick gene seek, and then we can get a Drosophila genome browser view that's showing us the location of that particular gene. If I scroll down here, we can bring that up here. We can see we've got this RPS5A gene on the Drosophila genome. Now, one thing you should notice here is that there are a lot less options. Typically, the annotation is not going to be as good for some organisms as others. Even a really well-studied organism like the fruit fly, generally we're not going to have as many resources and as much annotation available as we do with the human genome. And we can see more of that by picking, let's say, the maps and options on this genome. And if we go through some of the options available here, we can see there are a lot fewer things that are annotated to this. We can pick some of these, so I'll pick RNA-DM, that's adding an annotation for Drosophila melanogaster RNAs, and I'll pick a Drosophila cytogenetic map here. So if we add that in, we can add a Drosophila ideogram. Click OK here. And then just to make things a little easier, once this comes up, I will zoom out to the full chromosome view. And then we can see how things are maybe a little different on a Drosophila. So here we've got, on the right, a Drosophila ideogram. And you might notice that this looks very different than the human ideogram. It's used for a similar purpose to localize things on the genome, but it's a completely different technology for creating this ideogram, completely different nomenclature for referring to locations on it. So here we can see Drosophila cytogenetic coordinates look like 5b4 to 5b6, just as that community has their own way of referring to locations on their kind of ideogram. We can also see annotation about the different transcripts available. In general, there aren't going to be as many for a model organism as there are for a human, and it will depend on the organism we're looking at. Some transcription studies are very mature. There's a lot out there, some less so. Drosophila, we would expect to be actually very good in that regard. There's a lot of transcript data available, but still it's not comparable to what we see for human data. So that basically is what we would see in a model organism. But there's one other key thing I want to point out that's different between the Drosophila page and the human page, and that is this little link here that says FB. So for all those reasons I've been telling you that model organism communities often do things their own way, one of the things they often do their own way is create their own genome resources and their own genome databases. And the FB here is referring to an alternative for this kind of analysis, where instead of using the general NCBI tools, we might use a different tool for what's called the flybase. So flybase is basically the genome resource specifically for Drosophila. It's the place the Drosophila community goes to do all of the kinds of analysis I was just showing you that we might do in the Map Viewer. Right, so this is going to have its own pages. They're roughly equivalent to NCBI Entree Gene pages. This is an example of that. Not showing the detail here, but basically there are all these other kinds of information available. And some of this gives a hint as to why we would want this kind of specialized database for flies. So there's a lot of kind of information that is important to some communities and not important to others. So, for example, if you are doing studies of where genes are active, you have very different kinds of anatomy and very different kinds of stages of development in a fly than you do in a human. And so a general purpose tool is really not going to do a very good job. So you would end up with a fly version of this where you could get fly gene expression data, and that's going to be annotated in various ways, where you get special kinds of information that are specific to a fly that really would not be as easy to get in a human being. This is just showing some examples of that. We can also get things that you obviously can't get in a human being. So for example, we can go to stocks and reagents, and we can say, if we're interested in this gene, maybe we want to see a fruit fly that has that gene knocked out. So we can go to stocks and reagents. We can see all the places that maintain stocks of genetically modified fruit flies, and we can pick among all the options that have known mutations in this gene. And we'll see later in the class how we would look into these various mutant alleles, see what the phenotype is, and maybe pick one up. And then if you worked in a fly lab, you could call up the Bloomington resource and say that you want the fly strain that has this particular allele in it, and they'll send you your fruit fly. So that is the kind of thing that would be very community specific, and it's the reason why we need these specialized kinds of pages. Any questions about that? Okay, this does create some complications if we need to work with these resources. So, for example, this has its own genome browser integrated with it, this gbrowse. We click on that, we can see the genome browser used specifically by flies. We can see this RPS5A gene. Here's the annotation of different transcripts known with this gene. And it has its own interface, completely different than the Map View one. We can pick select tracks here, and this will have another way we can decide what kinds of annotation we want in that genome. So maybe here we're interested in removing the transcripts, but we want to see evidence behind these transcripts, so we'll turn on the evidence. We'll turn off information about mutations, and then we can just kind of go through and decide which annotations we really care about here. And after picking a few of these, maybe we decide that we've got all the information we care about. We can go back and see. Go back to the browser and then see maybe the evidence supporting the particular gene annotations we care about. So this is showing, in this case, a way we could view the annotation for all of the gene forms known for this particular gene, and we could use that to decide if we really believe this particular transcript, if we think there may be alternative splice forms that aren't annotated that might be important to us, and so forth. And so this is one of the things I would say when you get into recitation, when you try using some of these things, play around with some of these annotations. There are a lot of options. Some of them we'll be covering this week, some of them we'll be covering in subsequent weeks because they're relevant to later topics. But a lot of them, it's pretty easy to just click a box, see what shows up, and try to figure out what you're seeing there. So I'd say play around with it and see what's available. There's a lot of useful information, a lot more than I have time to go into here. Are there any questions about anything at this point? All right, so this Flybase is a really fully integrated database. There's a lot more to it than I'm showing here. We could find other genes, other kinds of annotations. There are a lot of other tools linked to it. So we can search for Heartless, a gene that you may remember from earlier in the class. We can get a page for that gene, see various other kinds of tools for analyzing it. Again, there's a lot more there than I have time to go into. There are also, of course, a lot of other model organism databases, and I don't have time to show you all of the important databases. One other one I'll show you, because it will come up quite a lot, is called the Mouse Genome Informatics site. So that's at informatics.jax.org. I'll just show you here the links if you want to go directly to these for Flybase or Mouse Genome Informatics. So this is the equivalent of Flybase if you work with mice. So I mentioned these two specifically because if you're doing research here at Carnegie Mellon, there's a pretty good chance you're either working with flies or mice. So if you're doing neuroscience, for example, you're probably working with mice. This is a place you would go to get all your information about mice. And just like the Flybase, this is something where we can search for genes. I'll type in HBB. That will bring up some classes of genes that might be of interest. And I'll pick one version of a gene that hit to that, the HBB1 gene. So that is the hemoglobin beta chain, the nature adult one. We can click on this, and we can get the MGI version of the equivalent of an entree gene page. So the set of information that the mouse community considers particularly important for knowing about the gene. And a lot of it is going to be the same sorts of things we've seen in other contexts, just organized in different kinds of ways. So some of this, again, we'll revisit later in the term. So next week, we'll be looking at issues related to mutant alleles, so variant alleles, and how those affect phenotype. Later, we'll be seeing information related to gene expression. And that would be another case where it matters quite a bit that we're looking at a mouse. You would have different kinds of expression data, different ways of looking at it. For the mouse community than you would have for the human community or the fly community or the sea urchin community or whatever else you're interested in. So I'm not going to dwell on this for today. We will be revisiting MGI and some of these other sites quite a bit later. But again, this is a good thing to look into. Just try some of the options available to it. Try out the mouse genome browser. So if I click on, click on the search here. I may be zoomed in too much to bring this up. You can find a mouse genome browser. It has its own version of an integrated genome browser, and that would be something you can play around with, learn to use, and learn about the different kinds of annotation that are available for a mouse genome that would be particularly relevant if you're doing mouse studies. Any questions at this point? Okay, so one additional thing I wanted to demonstrate is that even for a lot of people who need to use these general purpose tools, so either you're looking at human or you're looking at a new organism that's maybe not so well annotated, it doesn't have its own resources, a lot of people actually prefer to go away from the NCBI resources for this. So the NCBI Map View is really nice, and that's integrated to all of those other NCBI resources. But a lot of people like what's called the UCSC Genome Browser. It's called that because it's based out of UC Santa Cruz. And if we go to this web page, genome.ucsc.edu, and click Genome Browser, we can see how the UCSC Genome Browser works. I think a lot of people would consider this one a little easier to use, a little more straightforward. And we can see that by taking a particular human assembly. I'm going to stick on one that's not actually the most recent, because the most recent one is not as well annotated as the somewhat older one. And I will type in a number here, 17Q12. So what does 17Q12 mean? Yes, it's a cytogenetic coordinate. It's referring to a region of chromosome 17 on the Q arm specifically. So we can do a search for 17Q12, and we can see how the UCSC browser would present from 17Q12 to us. So we can get a region of chromosome here. That's a pretty large amount of sequence. So if we look specifically at the bases here, we're looking at about 7 megabases, so 7 million bases of sequence. And as we can see here, we've got a lot of things annotated. These would be transcripts. We've got a lot of variants of each of the genes here. And we might be interested in cleaning this up a little bit so we can see things that are particularly interested to us. And one of the things you should be able to see here is that there are a lot of options integrated with this. Each of these is representing a class of annotation track, and usually this gives you a number of different levels of detail for seeing an annotation track. So maybe we're interested in a dense form of UCSC genes. That would be a relatively sparse way of representing them, or a very compact way. Maybe we want to hide the rep-seq genes. We might not be interested in publications, so let's get rid of those. RNAs, maybe we are not interested in looking at RNAs for the moment, so hide those. Regulation, let's say we are not really, well, maybe we'll leave that in. Say we might be interested in seeing that. Here we just have 2 alternatives. And conservation, maybe we're particularly interested in. We'll keep on the full level of detail for the conservation track, and I'll add a squished kind of medium resolution view for this 46-way conservation track. And we could keep going here, going into all of these. I'm not going to dwell on them. I'm just trying to hide ones that will take up a lot of space here. And we could click refresh, and that should bring up a re-annotated version of this genome. And we can see some of these bits of information important to us. So we can see that we're looking at a small number of genes here. We can get information on predicted regulatory elements in these genes. So this ENCODE annotation is a way of trying to find these regulatory elements by looking for conservation across many organisms and looking at detailed data on expression and conservation and various other things. And we can look more directly at this kind of genome conservation by seeing tracks showing similarity across different organisms, similarity across vertebrate genomes, and so forth. And we could try maybe zooming in, so I'll zoom in 3 times, and then zoom in another 10 times, and maybe that will get us to the point where we can start looking at a particular gene. Another 3 times zoom here. And then we can start to see interesting features emerging for looking at one of these genes. So we can see, for example, that there is a piece of gene annotated here. We've got some intron and exon structure. And as we can start looking at these different tracks of annotation, we can see information about regulatory sequences associated with the promoters of these genes. We can see the conservation information, get information on how things tend to be conserved, maybe when we get closer to exons, less conserved when we're in introns. Basically get an idea on a relatively fine scale for a particular annotation of that gene. So if we looked at this gene here, we could see that we have these spikes of conservation, and they seem to line up very nicely with exons of those genes. That's more or less something we would expect. And again, you can see the same kind of conservation in the comparative genomics here. So we can look at very high conservation, with, in this case, rhesus monkey, breaking down as we get farther and farther away evolutionarily, until we get to the point where with fairly distant vertebrates, we only see conservation around the exons. So this is the kind of analysis we could do if we really want to know in detail what's going on with a particular organism like this. Any questions at this point about how we would use these tools, how we would interpret them? Okay, so again, there's a lot more to see here than I have time for, but I did want to show you that because when you work with these genome tools, often this will be an easier one to use, and we'll have a lot more kinds of annotation available if you might be interested in. So there are a couple more things I wanted to show you in the remaining 10 minutes or so of class, and that refers to some of the resources for looking at smaller genomes. So often we're really interested in things like a human being or a model organism, but there are a lot more genomes available for particularly small organisms, so prokaryotic genomes, viral genomes, and so forth. And the tools tend to be somewhat simpler for those, but I do want to show you how that works. So I'm going to go back to the Map View page. This is one of several ways you can find these resources. And I'm going to start by moving to the bottom here and picking bacteria. So the small genome page. We get some information on microbial genomes. I'm going to pick prokaryotic projects here at the right, so we can see about information on prokaryotic genomes. We get a page devoted to all the different prokaryotic genomes available or in process. You can see there are quite a lot of those. I will pick prokaryotes here, and I will limit this to just complete genomes to narrow it down a bit. And maybe limit this by picking a group here to bacteria. So throw out the archaea here. And then we can see all the different options available, more than 3,000 of them, of genomes of prokaryotes that are available for search or analysis in our data resources. If we just pick one of these at random, so we can start with this one at the top here, a Caryopleurus marina. This we can see is a cyanobacterium. Does anyone know what a cyanobacterium is? It's a photosynthetic bacterium. These used to be called blue-green algae, but basically they're bacteria. We can look at it, this particular genome. I may need to zoom this out a bit to be able to get to what we're interested in here. And we can see that there is some information available on replicons within this genome. So those would basically be the sequences associated with this genome. And if I pick the chromosome here, that would be the major chromosome found in that particular genome. And for these small genomes, they're typically going to be GenBank pages devoted to a single genome. So this is the GenBank page devoted to the complete chromosome for a Caryopleurus marina. And all the tools we had available for looking at a GenBank page would be available to us for looking at this kind of genome. So we could customize the view here. I will say that we are interested in, say, gene, RNA, and CDS features. We won't show the sequence because the sequence would be several megabases. But what we could get out of here is information on every single gene annotated in that genome, what the annotation associated with it is, or if there's a name associated, or it's just a hypothetical gene, where it is on the genome. And of course, we could add in the complete genome sequence. So if you were working with this more from the point of view of developing your own analysis tools, it would be fairly easy to download this whole GenBank page and write things that would do whatever parsing you need to do. So find the genes of interest to you, do analysis on the sequences of those genes, on the translations, whatever. It's fairly easy to get that kind of information out. Often we're interested in a more manual analysis though, so I'll just show you. If we scroll back to the top here, we can pick a graphical view. And for a bacterial genome, generally what we'll be using is something much more like the sequence viewer. In this case, this is actually exactly the same viewer as we saw for looking at finer scales within the human genome, so the NCBI Seq view. And we can get an appreciation for what a bacterial genome looks like by zooming this in a bit. And we can start to see all the different genes within a region of genome there. So here we're looking at a small piece of genome. This is about 90 kilobases of genome. And each of these red things is showing you an annotated gene on this particular organism's genome. And one of the key points I wanted to illustrate here that you should be able to get from this is something I had mentioned in the previous class, that it's just covered with genes. And this is typically what you will see in a prokaryotic organism. There's not a lot of wasted space there. Almost everything in the genome has a gene on it, and that you can see that pretty clearly here with all of these different genes annotated in this relatively small piece of sequence. As is often the case, there are a lot of other resources integrated into these. So we could mouse over a gene here and get links to a GenBank page for that particular gene, various other ways of getting information about it, blasting it, looking at retrieving the sequence, whatever. Again, you can play with these things. I don't think I need to explain in detail how they work. Any questions about that? One last thing that I think is neat to see if you have never seen it before is if we go even simpler, look at a viral genome. So we'll go back to Map View again. We had near the bacteria here, we could choose viruses, go to the viral genomes, and I will pick Browse Viral Genomes by Family. We can see that there is a large taxonomy of different classes of viruses we can choose from. I will scroll down here, pick one particularly interesting kind of virus called a retrotranscribing virus, pick the complete genomes, and we can see that there is a fairly large category of genomes that will come up for retrotranscribing viruses. We have a number of plant ones, and I'll scroll down to try to find an interesting human one. So we have here the Retroviridae, and if we scroll down, we can find, let's say, human immunodeficiency virus 1. That's a pretty medically important one. And we can go to the GenBank page for that genome, and we can pull up a canonical human HIV-1 genome. So this is, again, a GenBank page, same kind of thing we've seen before, and it's annotated, in this case, with a more expanded view. So we're seeing all the locations of all the genes, we're getting translations of all the genes, and in this case, we can actually get the entire genome sequence. So I'm scrolling through it here, but if you saw that go by, that was the HIV genome. It's about 9,000 nucleotides. Doesn't sound like much for something so important, but that is the entire HIV genome. Has, depending on how you count, about 10 different genes in it, and we can really study that in detail. We can look at great detail at every particular gene in that, or we can bring up a graphical view. And again, I think it's amazing how simple these things are. This is, again, the entire HIV genome. So we see 10 different genes here. Again, it depends on how you count, because viruses tend to be quite complex in the way their genes work. We can have overlapping genes. Sometimes we'll have a gene running in one direction on one strand, the other direction on top of it on a different strand. So these tend to have these very compact genomes. And we can get a very fine-scale view of what's going on in the genome like this. So just like we've seen before, really look in fine detail, even at the level of sequence, and see exactly what's going on at any given point in this kind of genome. And that's the kind of detail and the kind of resolution you can get when you're working with something really simple, at least in terms of sequence, like a typical virus. So any questions about any of this? Okay, so again, I know there was nothing particularly deep about today's lecture, but there are a lot of useful tools to be aware of, and I hope it's useful to you to see a demonstration of how some of them work. But really play with these in class, working on your recitation assignments and working on your homework assignments. There's a lot more to learn, and sometimes you just have to try it on your own to do that. Are there any last questions before we break for the day? If anyone hasn't turned in your homework, please make sure you have that here.